So, in 1983 Capstan Sander, right, they developed a program called the DSSP, right. This is dictionary of secondary structure of proteins, this is they called as DSSP, to analyze the known 3D structures. Here they are not doing any prediction, they are taking the real structures from the protein data bank, and they estimate they just check the head and binding pattern, and then they have the 5 angles. And from these patterns they assigned each residue to 8 different secondary structures, right, 3 different types of helices, right. What are 3 different helices we discussed?
310
on 5 helix, 310 helix and 5 helix, and 2 types of strands, right, and the bridge, and 1 bend and 1 turn, right, and 1 coil, one is irregular. So, they make 8 different categories of secondary structures. So, here this is the output of the DSSP, okay, this output of DSSP. Here they give the chain information, this is the chain information, and here this is the amino acid sequence, and this is the secondary structure assignment. And here they have these head and binding patterns. If it is the residues number, they put 2, 4, 1, minus 1, that means plus 1 means the forward direction and the minus 1 will be the reverse direction. They put which residues are head and bonded with respect to this particular residue, right, and they give some energy values also, this is what they give the energy values. Then finally here they get the phi psi values, this is phi, this is psi, right, you can see the almost the residues are within that range, right, if you see this range, right, and here if you see the map, okay, here this is the range, right. So, this is the angle, right. So, you can see the values are within that particular range. So, you can see this is the from the alpha helix. So, now the question is, if you have this primary sequence, right, DSSP can assign the values if the 3D structures are known, right. This is one example for the myoglobin. First line it shows the sequence, and second line shows the secondary structure. If the structures are not known, for example, this is for example we give the sequence, is it possible to predict the secondary structure from this sequence? From this sequence where there are alpha helices, right, this alpha helix, where you can form the beta strand. So, there are various methods, because once we have the data, the sequence and the structures, then they can compare the results, right, sequences and the structures, and based on the information you can derive some methods to predict the secondary structures, and you can evaluate, because there is some experimental known data are available. So, you can also evaluate. So, there are various methods which have been proposed for predicting the secondary structures of proteins. The first one is based on statistical analysis, this is the simplest one. For example, alanine prefers to be in alpha helix, then if you see few alanines, then we see that okay, this segment prefers to be in alpha helix, right. For example, if you see the students, they will come at 9 o'clock, they go somewhere, 10 o'clock come to the lab, right, and 1 o'clock go to lunch. Then if you see his behavior, where he will be, for example, 10 o'clock, okay, he will be in the lab, it's 1 o'clock, he will be in the lunch, right. Likewise, if some residues they prefer to be accommodated in any specific secondary structures, then we can see that okay, these residues are highly populated. So, this segment forms an alpha helix, right. This is called a statistical analysis. Swayam Prabha they analyzed the data of the then known proteins, all the known proteins available at that time in 1974, and they developed the propensity values for all the 20 residues. Based on that, they classified the residues into few groups, which residues prefer to form alpha helix, which residues they do not prefer to be in alpha helix, right. Some of them which are indifferent may or may not form, right. Likewise for the beta sheets. Then after this information, this you can achieve an accuracy of about 55 to 60%. Sometimes same residues they can adopt alpha helices and beta sheets, right. The prediction method can predict either helix or strand. If they prefer same segment in 2 different secondary structures, then one will be correct, one will be wrong. In this case, Garnier's group, right, they developed a method called GOR based on information theory. Here they did not consider only a central residue, they made a window of 70 residues, right, 8 residues at the left side and 8 residues on the right side, right, and get the information for all the residues. That means any segment they have different secondary structures, it based on the neighboring residues. So, you can discriminate if the same segment belongs to helix and belongs to alpha beta sheet depending upon the neighbors. So, they use that that information to predict the secondary structures. Later on they try to use various properties. One of the most prominent properties is the hydrophobicity, because it can easily distinguish the residues based on polar, non-polar, charged and all. So, they try to see the periodicity of these residues. They take the sequence and find are there any periodicity of residues in the sequence, right. If you look into the sequence, difficult to find. So, if we have a kind of figures, kind of profiles, easily you can see, right. What is hydrophobicity profile?
Sequence versus
So, plot connecting sequence versus values, right. So, we discussed about several patterns, right, for alpha helix, for the strand, for the transform regions. So, they have some specific patterns, and if you scan these patterns in these profiles, right, and then you can easily identify the secondary structures. Then after that they try to include the multiple sequence alignment. So, right, the time grows, they think the single sequence it will be difficult to predict the secondary structure. If you have more number of sequences of known information, then you gather the information from that sequence alignment and you can try to use for prediction. So, earlier we discussed about multiple sequence alignment. How many residues are required for the multiple sequence alignment? 3 or more sequences. If you want to see any residues are conserved, how many sequences can give the reliable results?
100.
Yeah, if it is more than 100, right, at least we need 50. If it is more than 100, then you can get reliable results. So, in this case they try to gather the information for different sequences and make the alignment, and from this alignment they try to predict the secondary structures, right. For example, position number 5 accommodated highly conserved accommodated by alanine, and in all the 100 sequences this residue is in helix. Then your own sequence you can also tell that this residue number 5 alanine that belongs to alpha helix with the high confidence, because it is highly conserved. So, this program depends on the sequence alignment. If you get good alignment, then your accuracy is high. If the alignment is not very good, then you have the dilemma in your assignment. So, then we used multiple sequence alignment, then we discussed about the profiles, pieces of matrices, right, and then they included all the aspects to predict the secondary structures. Then they started to use machine learning, because all these things, right, depending upon the data sets, right, we need to manipulate the data, right, write the codes, and they try to use different machine learning techniques, for example neural networks or support vector machines, and then see we train all the known information with these machine learning techniques and to predict these secondary structures, either helix or strand or coil. So, more details we will discuss in the next subsequent classes. Then during this course, right, they started to use join methods or a consensus method, right. This is easy to do, but also it relies on different servers, different methods. In this case, either they use for voting. For example, if you get 10 methods available already, because based on statistics or information or whatever, right. So, different methods are available, then they run for example 10 servers, right, and see the best. For example, 7 methods predict a residue as helix, then you can say this residue belongs to alpha helix, right. Just by voting procedure you can predict the secondary structures and you can evaluate. Second one is you can get the output from different servers and you can make a meta server. You can again run this output, right, train this data to get this prediction results for your sequence. So, current scenario they try to mix everything, they try to include as much information as possible, either from single sequence or the statistical preferences or the multiple sequence alignment, the matrices, right, and put together in machine learning, right, to get the secondary structure prediction. Currently they are using the deep learning, right, because it can handle large amount of data. So, in that case you can enhance the prediction performance. So, then we will discuss about the statistical methods. The statistical methods, first we need to have the preference of residues in any secondary structures, either in helix or in strand. How to calculate the propensity? So, what is the meaning of propensity? Propensity of any amino acid, for example alanine, right, that depends upon the preference of that particular residue in any particular conformation. For example, if you take alanine or any residue I to be in any conformation, for example helix or strand, right, how to do it? First see the percentage of the residue I in that conformation compared with all the residues in the same conformation. So, consider the propensity of I using the equation percentage of residue I in alpha helix divided by the percentage of all residues in alpha helix. How to calculate the percentage of residues I in alpha helix?
Number of ith residue in alpha helix divided by all residues.
All the residues of type I. For example, alanine, 10 alanines, right, and 7 in alpha helix, right. What will be the percentage of residues in alpha helix? 70%, right. So, then you can see that. Then normalize with the total number of all residues in alpha helix, the whole protein, 100 residues and 80 residues in alpha helix. Then how many residues in alpha helix? 80%. Now you compare. So, if you whole protein, if 80% alpha helix, and the preference is high only if it exceeds the number, right, because if it randomly distributed you can get 80%, right. Then you can see whether it is underestimated or overestimated, then that will introduce a bias and you can see some preference of these residues in alpha helix. You can say percentage of residues in alpha helix using this equation n alpha by n of I, where this is the ith residue say alanine in alpha helix divided by ith residue in the whole protein. Then likewise you can the whole protein is n is the total number of residues, n alpha is number of residues in alpha conformation. We will discuss with one example. Here first one is the sequence and the second one is the secondary structure. So, you can see H is a helix and you can see the G is 3-turn helix, right, and you can see T is turn and you can see some gaps. Gaps means you can say it's coil because that is not known, this is irregular shape, right. So, this is the S is the bend, right, and you can see the different assignments using caption standard, they use the DSSP. So, this is the result of the DSSP. Now, if you want to see the propensity, for example, if you take this protein, what is the propensity of alanine to be in alpha helix? How to calculate? So, you can see alanine in alpha helix, right, this is number of alanine in alpha helix divided by number of alanine. So, this is total number, how many alanines here? You can see all the alanines. Three, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. There are 16 alanine. Out of 16, how many of them are in alpha helix? If you see the red ones, they are 15 here, right. So, this is not alpha helix, right, others are alpha helix. So, there are 15 alpha helix. So, you can get the numbers, there is 0.94. Then take the all the residues in alpha helix. In this case, you can see 153 residues, right. If you see here 50, 100, 150, 153. Out of 153, you can see 115 in alpha helix. So, there is 0.75, 75% of the residues in alpha helix. Now, if you see any residue to be preferred in alpha helix, that should be more than 1, that should be more than 75% because the whole protein 75% alpha helix, right. So, then you can get the propensity of alanine. So, 0.94 divided by 0.75, this is equal to 1.25. So, the value is more than 1, then you can say that this residue prefers to be in that particular secondary structure, in this case for example alpha helix. Then you take the glycine. If you see the glycine, how many glycines here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 9 or 10 glycines, right. So, finally, you can see this is the 0.5, the percentage of glycine, this is the percentage of glycine and this is the percentage of helices, right, this is 0.75. So, divide this one, they will get 0.66. So, in this case, this is less than 1. From this, what can we infer? Glycine is not preferred in alpha helix. This we did based on just one protein. So, currently more than 130,000 proteins are available in the protein data bank. You can reduce redundancy, right, and from the non-redundant set you can calculate the propensity, then you can get a value for 20 different amino acid residues. Then how to derive, how to get these numbers, right, if you want to derive these propensity values from the whole database, right, how to get the numbers, what is how to write an algorithm to get these propensity values, what are the input information do you need?
Sequence.
So, we need the sequence, right, on the 20 different amino acid residues, right, and you need the secondary structure assignment, you need the sequence and you need a secondary structure assignment, right, and for 20 different amino acids. First we need to get the occurrence of 20 residues in helix and we need the same occurrence in the full protein. So, full protein how many residues of a particular type, each type we can get, and how about in helix, then you can calculate the ratio. This will give you a percentage of residues type I in helix, and then the second step you take the total number of residues in helix and total number of residues in a protein, that is n alpha by n, right, this will give you the percentage of residues in alpha helix. Then you divide this by this, then you can get the percentage of all the 20 residues in helix. You can repeat for all the proteins and finally you can get the propensity values. For example, if you take the helix, okay, this is the propensity values, this is the data obtained from Chowan-Fasman. If you see these numbers, some of them are more than 1, some of them are less than 1. Based on these numbers, which residue is preferred to be in alpha helix? Glue, alanine and leucine. Which residue is not preferred to be in alpha helix? You can see proline, glycine and so on. Okay, this is the have high preference, right. So, these residues are no preference, right, less preference. Then if you see the values, some of them are very high values such as 1.5, 1.45 and some of them are very low, for example 0.5 or 0.6, and several residues they are oscillating around 1, right, that also depends upon the data set. If you use 100 proteins you can derive the values, if you use 1000 proteins or 10,000 proteins, right, although there is not major difference, but you can see a similar trend, right, maybe some minor changes. Hence they divide this group into 6 groups. First one is capital H alpha, this is strong helix former. So, because they have high tendency to form helix because the value is very high, and they have the small H alpha, right, that is in this group. So, they have a tendency to form H alpha, right, to form the helix, that is helix formers. Then here just around 1, little bit around 1, here this is weak helix former. Then come down, there is just a little bit less than 0. So, they make it as weak helix former, helix breaker because it does not prefer to be in the helix, and you can see B alpha, this helix breakers, and capital B alpha, there is a strong helix breaker. So, if you have the propensity based on the numbers, they grouped into 6 groups: strong helix former, weak helix former, weak helix breaker and strong helix breaker. And you can use this information to predict the secondary structures. If you have any segment which contains mainly these residues, then there is a high probability to be in helix. If they have more residues which are in this category, then it has less probability to be in alpha helix. This for helix. Then we can do this for beta strand or coil or turn. If you see the beta strand, right, here are the classified 6 different groups, and you can see the high values. This is the strong beta formers and these are the beta breakers. And if you compare this P alpha and P beta, some residues are distinct and some residues are same. For example, proline, right, here it is not preferred in alpha helix, also it is not preferred in beta strand. Glutamic acid, it is a breaker in the case of beta, but if it is a strong helix former in alpha. So, if you have a glutamic acid in a sequence, then you can see there is a high tendency to be in alpha helix, right. Likewise, some different preference you can see the preference in beta, but not in alpha. But some cases you can see similar preferences in both alpha helix and beta sheet. This will give you a conflicting situation, right. The same segment can be predicted with alpha as well as for the beta. In this case, you can compare the values, which one has high values, then you can assign accordingly. So, how to predict the helical segments or beta strands, right, from this amino acid sequence? First, you see the segment, the sequence, and assign the propensity values, because if you take this table, there are 12 different groups for the strand helix 1, 2, 3, 4, 5, 6, right. For the beta also you have 6. So, you can take these preferences and assign the values. then you can see the weightage. For example, if it is helix, so we take the different parameters, for example H alpha, this is the helix former, we take it as 1, and this is the weak helix former, take it as 0.5 for example, this is the weak helix breaker, they take it as 0, and this is helix breakers, they put at -1, because we have to give preference for the residues which have high preference to be in alpha helix. If it has no preference, it has less preference, take it as -1. Then you take a window residue of 6 residues, because alpha helix is longer than the beta strands, right. So, take the minimum 6 residues and you can extend it, and the idea is if you take any segment, this segment have high preferred residues to be on alpha helix and less number of low preferred residues. So, at least 4 helix formers, and there should not be not more than 1 helix breaker. So, with this assumption they put a score of more than 4. In this case they will have high tendency to have at least 4 helix formers, because we take the value of 1 and not more than 1 helix breaker, right. In this case you can have a score of 4. Then you can extend the directions. For example, if you find, if this is the sequence and you find okay, this is the helical segment, and then you can extend and see whether the score is down or not. If the score is still more, for example the preferred residues are present together, then you can add up, and then finally you can add up to here to make the complete helix. So, now how to do this? To continue the search, and there are some conditions we can assign the terminals. For example, you can see some residues which are mainly in the N terminal, some of them on the TC terminal, and proline, as we discussed, is not present in the inner helix because it will form turns. These are the some common observations. This you can use for refinement. If you have more residues are predicted, right, and if you want to refine it, then you can use this information. So, now I give a sequence. Okay, this is the amino acid sequence. So, now the question is which residues form alpha helix, which residues belong to beta strand, right. First take the sequence and we take the overlapping residues of 6 residues. So, 1, 2, 3, 4, 5, 6. So, KBF GRC, this is segment 1, and the second one go from here to here, right, VFGRCE, and the third one is start from F 4, 5. So, likewise you can make different segments. Now, you take this one and see the score, right, instead of just writing the number, just we check whether this is 1 or 0. So, for example, this is the table, okay, you can see this is 1, okay, this you put 0.5, this is 0, this is -1. Take the first one, K, you take K, this equal to lysine, okay, this equal to 0.5, okay, lysine is here 0.5, right, then V is here, this is equal to 1, then F is here again phenylalanine, so this is equal to 1, right, and this G is in this group, this is -1, so we put the -1, and R and C, that is here, R is here and C is here, this is 0. We assign the values, just approximately assign this 2.5, so this is less than or equal to 4, right, this is not the part of helix. Go to the next segment, here, right, here these numbers are the same, and the last one is E, that is equal to 1, right. In this case if you add this equal to 2 or 3, 1, 2, this should be 1.5, right, 1, 2, -1, 1, this should be 1.5, right, this 1.5, right, this will be 2. Now, the third one if you see, so you have these are the same, right, these are the same here, and the last one is L, leucine, this equal to 1, this equal to 2 again. Go on adding these numbers. When you go with RCE LLA, R equal to 0, C equal to 0, all these are 1, because E is 1, L is 1 and alanine is 1. So, final score is 4. Now, you can say this is a segment, you can identify as an alpha helix. Now, whether this only this residues form an helix or you can extend it. Okay, then if you take the last one MKRH, it is here, and then if you do it here. So, if you do this 4.3 you can add, right, this is RCLLA, now this is next A. If you add the A is preferred one, so it will be more than 4, right. The next one is M, so M is also preferred one, this is belongs to H alpha, right, M belongs to H alpha, so that also you can increase it. So, next one is K, so lysine, so it is also 0.5, and if you add the values, right, this is, then this will give you the value more than 4. You can extend up to this mk rh. So, here you can add this m is equal to 1.2 right from here, and k is 1.07, 1.07 lies in here, and arginine 0.79, histidine 1.24, this value is 4.3. Then add next one, if you add one more krhg, right, you can see the last 4 one krhg, right. If you add this, then the number is down less than 4. So, you cut here. So, now segment starts from this lysine, right, you can start from here this one, right, you can, right, where it is 4 here, it starts from R, right, you can start from here and up to here, right, this is the segment which can form alpha helix. So, how to do this? First what to do? 6 residue windows, you get the overlapping segments. Okay, these are the 6 residue overlapping segments, and then
see the score.
Then get the assign the values from this table. We went to 6 different categories based on the tendency to form helix, or they are not preferred to be in helix. They have -1 to +1. Just put the numbers, assign the numbers, and see whether this number is more than or equal to 4. If it is less, go to next segment and go to next segment up until where we get the value of 4. Right, you get the value of 4 here. So, we can start from here, then this is 4. The question is, if you add 1, that will have the high preference or low preference. So, add 1 and see the last 4 residues and whether the number is right, you can see the segment, the 6 segment value is more than 4. So, you can see the numbers, right, this 4.3, because you can get the numbers from this table. Here we just assign the numbers 1 or -1 0.5 just to see how many helix formers or how many helix breakers. Once we identify the segment, then extend it based on the real values, right. So, then we extend it until we get the value less than 4. Here you can see get the value less than 4, then we stop there, right. This is the complete segment you can get. Okay, so, this is assignment, and actually they compare the experimental data, it also, okay, it is up to here, right, this is completely matched with the experimental data. This is a segment, here we could not get any helix, values are less, and here we get the high preference, and the segment is predicted as alpha helix, right, it matches with the experimental data. Now, how to do the beta strand? Okay, do the same procedures, take the h beta small h beta equal to 1, I beta equal to 0.5, small I beta equal to 0, and capital B beta or B beta equal to -1. So, here we use a scan of 5 residues and set a score of more than 3, because all beta strands are smaller than alpha helices. So, take 5 residue segments, right. How they define these segments? They tried with various segments and compare the results with the experiments, and finally which one matches they try to fix with that one. This is how they assign 6 and 5 for the helices and strands. Then you can extend the length in both directions unless the average property is less than 1, right. You can continue the regions and find the strand. Here the same procedure you can adopt, so I do not have to explain all these details, right. So, here we have the values for P beta, and you can assign the values and find out. For example, if you find a conflict situation, right, same segment can be predicted as helix and can also be predicted as beta sheet. In this case, what to do? In this case, get the segments and assign the real values and see which one is dominant. If average propensity alpha helix is greater, then this is alpha helix. If beta strand is greater, then it is beta strand. So, in the conflicting situation, you can get the values and you can compare the numbers and then assign the secondary structure. So, here this is one web server. There are several servers available to predict the secondary structure based on Swayam parameters, right. Here they use various programs, and here we take the Swayam parameters, and here we give the sequence in FASTA format. It uses this method, the same 1974 Swayam, right. And finally, you can see this region is the helix, this is what we discussed now, the predicted ones, and then you can see this is a strand region and this is a turn region. So, finally, you can use this server to predict the secondary structures based on Swayam methods. So, summarizing, what did we discuss?
Protein secondary structures.
Protein secondary structures. What are the various secondary structures?
Helix.
Alpha helix, beta strand, right, turns, right, and coil, right. How the secondary structures are formed?
By hydrogen bonding.
Many hydrogen bonds between backbone NH and CO groups, right. So, they have the periodic arrangement of residues, right. So, alpha helices are how alpha helices are formed?
Hydrogen bond between I and I plus 4.
I and I plus 4, different types of helices, right, and this is closely packed. If you compare to the alpha helix in beta sheets, right, this is closely packed and this is loosely packed, beta sheet is loosely packed. right. Then how we obtain this Ramachandran plot? What is Ramachandran plot?
Allowed and disallowed region.
Right, it's a plot connecting
Alpha
phi and psi, which tells you which rotations of phi and psi are allowed in different secondary structures, alpha helices and beta sheets, right, and which are allowed regions and the partially allowed regions, right, and the complete disallowed region, right, fine. So now how to assign the secondary structures? If you know the 3D structures, Capstone Center developed a program called DSSP. What is DSSP? Dictionary of secondary structure of proteins, right. They assign the secondary structures based on Herring bonding pattern, right. They descend the values, right. Now the question is whether we can predict the secondary structures from the sequence. Several methods we discussed, right. I will give the details in the next class, right. So mainly we discussed in detail about statistical analysis, that is Chau-Fasman method, right. So how the Chau-Fasman method works? Preference of residues. How to get the preference?
propensity value.
Propensity values, right. For any residue, how far that specific residue prefers to be in any secondary structure, either helix or strand. Based on that residue in helix, total number of residues, totally how many of type I, total number of whole residues in a full protein, and how many residues in helix in a complete protein. So based on this information, you can get the propensity. Can you tell one example which residues prefer to be in alpha helix? Alanine, glutamic acid, okay, is preferred to be in alpha helix. Some residues which are not preferred to be alpha helix, for example, glycine, proline, right, they are not preferred, right. So based on this information, how to predict the helical segments?
With Chau-Fasman method we can.
Right, you take a window length, overlapping 6 residues, right, and then classify these values in 6 groups and assign the numbers and see whether the number is more than 4, right. We continue till we get the value 4. Once we get the 4, that could be a nucleus, right. Then extend the residues and see the last 4 residues have the actual value of more than 4. If it is 4, add up, where it decreases, right, less than 4, then you stop. Then you can identify this residue is an alpha helix. Likewise for beta strand, right, they use how many residues? 5 residue windows, right, and then repeat the same procedure, right, to get the beta sheet. If there is a conflict situation, for example, same segment is predicted as both alpha helix and beta sheet, right, how to do this?
One with the greater average.
The one with the greater one, right, you can assign whether it is alpha helix have more propensity, right, then you can assign as alpha helix or beta strand vice versa. So now we can predict this using different servers, right. One example is you can see in this web server, right. So you can just give the sequence and give you the secondary structure. Then we discussed about the statistical methods, and next class we will focus on the different other methods. For example, the information theory, multiple sequence alignment, hydrophobicity profiles, the neural networks, right, like machine learning techniques, as well as the consensus, right, that we will discuss in the next class. Thank you very much.